Morgan to our platform to say he's doing a great job and I should definitely invite him. And I was actually go to his website and check through his publication, looking to the work. I was completely blown away. So I think it's really exciting to learn Morgan's work and then introduce to all the faculty and trainees on our platform today. Very, very brief introduction about Morgan. So Morgan did his undergraduate study in Department Molecular and Cellular Biology in Harvard, and then continued with PhD training in biological science in Rockefeller. Afterwards, he moved to Stanford to complete his postdoc training in both Department of Biological Science and then microbiology and immunology. From 2007, Morgan started his independent research program as a system member in immunology program in Sloan Kettering Institute Cancer Center, all the way up to 2017, become full member in Sloan Kettering. So Morgan's lab actually mainly integrates in imaging, synthetic chemistry, and material science to study how immune cells communicate, with a focus on the dynamic interactions that underlie immune responses. So immune cells like leukocytes possess remarkable structure plasticity, allowing them to rapidly recognize in response to receptor stimulation. This ability is central to their role in identifying and responding to infection and cancer. His lab investigates the signaling pathway, mainly regulate leukocyte architecture and how cellular structure influence immune function, aiming to inform a new strategy for modulate immune responses in vivo. Now, in addition, his lab also studied how immune cell communicates through transient structurally dynamic cell-to-cell contact, like immunological synapse, which regulate the transfer of information, and then also study the mechanism modulate these interaction, how they actually function in vivo during the cancer development. So as I said, Morgan actually really did a remarkable amount of work through his career, and then based on these, he was received numerous awards. I'm just going to list a few of their here, and then, for example, he received Serial Scholar Awards, and then Cancer Research Institute Investigator, Leukemia and then Lymphoma Society Scholar, and then also Baylor Award in Basic Research. So it's really exciting to have you on our platform. We are very much looking forward to your talk.
Thank you, Chuan. That was a very overly kind introduction. I'm very excited to be here, and thanks so much for the invitation. So as Chuan mentioned, my lab studies the structure and the function and the mechanobiology of immune cell-cell interactions. And over the last 10 years, we've been particularly interested in how the biomechanical dimension of these interactions contributes to their function. And so today I'm going to talk about an ongoing line of investigation in the lab that has to do with how lymphocytes use mechanical cues to regulate their responses to cancer cells during immunosurveillance. And basically, this we typically think of cancer cells being recognized by the molecular components on their surface. This is a somewhat different idea. This addresses whether lymphocytes recognize cancer cells based on how they feel. And so I'll sort of expand on what I mean as the talk goes on. So I'm going to be focusing on cytotoxic lymphocytes, cytotoxic T cells and NK cells. Cytotoxic lymphocytes, as you know, play a critical role in the body's immune responses to intracellular pathogens and to cancer. And they function by directly recognizing, physically engaging and destroying infected or transformed cells. And that recognition event is mediated by cell surface receptors that recognize key determinants on the surface of target cells. So in the case of T cells, we're talking about the T cell receptor, which recognizes cognate peptide MHC. In the case of NK cells, we're talking about receptors like NKG2D that are recognizing ligands that are associated with stress or dysfunction, like the MYC family of proteins. And these interactions are facilitated by adhesion molecules like the beta-2 integrin LFA1. Once target cell recognition occurs, it induces the formation of a stereotyped interaction between the cytotoxic lymphocyte and the target called the immunological synapse or the immune synapse. At that point, the lymphocyte kills the target cell using essentially a cellular venom that's composed of perforin and granzyme. And these toxic molecules are housed in secretory lysosomes called lytic granules inside the lymphocyte. And they move to the interface and release their contents into the intercellular space. Perforin is a pore-forming molecule that forms holes in the target membrane. And this allows granzymes, which are proteases, to enter the target cell cytoplasm by a number of different mechanisms where they cleave a defined set of substrates to induce programmed cell death. So the molecular components of this interaction have been known for decades. What's become clear more recently is that this all takes place within an intensely physically dynamic environment. And so to give you a sense of what I mean, I'm just going to show you a YouTube famous movie that was made by Alex Ritter, who was in Gillian Griffith's lab at the time. And this is of a cytotoxic T cell, a CTL, killing a tumor cell. And the CTL is expressing an actin probe, so you can see the cytoskeleton. And I'll play the movie, and you'll immediately appreciate how physically dynamic this interaction is. In particular, it looks like the CTL is burrowing its way into the surface of the target cell, and if all the estimates of this aren't really great, but it's been speculated that cells can use almost half of their ATP on actin dynamics like this. And so if all a T cell needed to do to kill a target cell was to stick to it and spit at it, then why waste all this energy on these actin dynamics? And so about the time we started looking at these movies, it became clear that lymphocytes can actually exert nanonewton scale forces across the interface with these actin dynamics. And so we became very interested in what these forces and mechanical properties in general could be contributing to the process. A large chunk of what we've done so far in this area actually focuses on what I like to call the efferent side of the response. So after the synapse is formed, how does the cytotoxic lymphocyte use force to potentiate the killing of the target cell? And so we found, for example, that force sensitizes target cells to perforin pore formation by straining and distorting the surface, and also that forces spatially guide the delivery of perforin and granzyme to specific regions of close adhesion within the interface. And that work is still continuing. But what I'd like to talk about today is the sort of an earlier step in the process where force contributes, actually the afferent phase of the response. It turns out that force and mechanical properties actually dictate the degree to which the cytotoxic lymphocyte becomes activated by the target cell. And the basis for this phenomenon is thought to be the mechanosensitivity of the receptors themselves that mediate lymphocyte activation. And so the integrins like LFA1 are sort of archetypal mechanosensitive receptors. Integrins famously have two conformational states: a bent state in which the ligand binding site is obscured, and an extended state where the integrin can now bind ligand. But this extended form of the integrin is not actually the high affinity, high activity form of the protein. In order to get optimal binding affinity for ligand and optimal signal transduction, you actually need to apply pulling force to the tail of the integrin. And this stimulates the formation of a catch bond at the ligand binding site. So it actually increases the affinity for ligand, and it also leads to the creation of these force-gated binding sites for downstream proteins on the integrin tail complex. that allow signaling to occur. And so in order to bind strongly and signal, you need to actually apply force to the receptor. And over the last decade or so, there have been indications that have emerged that other receptors like the T cell receptor, NKG2D, CD16 on NK cells, they are also mechanosensitive in the same way, and that they form catch bonds. And catch bond formation is a link to force-induced conformational changes on the cytoplasm side. These data are more controversial. The research around integrins is very solid. But what all of these lines of investigation actually imply is that there is a mechanical role for the target cell surface, because if you're pulling on a receptor to activate it, the target cell surface needs to be rigid enough to pull back, right? And so this has implications at the cellular level. So how this plays out at the cellular level, and this has actually been very well established, is that if you take activating ligands for a lymphocyte and you put them on a soft stimulatory substrate or a stiff substrate, like such as a soft hydrogel or a stiff hydrogel, the same ligands on the stiff hydrogel will drive stronger lymphocyte activation. And so this is an example from one of Claire Evro's papers. So here she took anti-CD3 antibody and she immobilized it on hydrogels of increasing stiffness. And you'll see that results in increasing cytokine secretion by the T cell. Right. And so based on this, one would expect that if you took a cancer cell or any other target cell and you didn't change anything about its cell surface, but you just increased its stiffness, that you would actually sensitize it to cytotoxic lymphocytes by making it more stimulatory. Right. And so that raises a couple obvious questions. The first is, do infectious agents in cancer modulate their biophysical properties to elude the immune system? And so this is something that we're quite interested in, and we have active projects looking at this. But the question that we actually ended up addressing first was sort of a corollary to this question, is does the immune system use altered biophysical properties to recognize dysregulated cells? And this is what I'm going to get to today. And the reason we started working on this question first is because a really great model system kind of walked into our lab through collaboration. And this started as a collaboration with Joanne Massaguet's lab on metastasis. And so the Massaguet lab, they're also at MSKCC, they focus on metastasis, and they've spent a lot of time characterizing this pathway, which is how metastatic cells colonize new organs. So it turns out that when metastatic cells move into new organs, they like to live on the abluminal surface of blood vessels, the outside surface of blood vessels. It's a great place to grow because you have access to all these bloodborne nutrients. The issue with occupying this space is that there are other cells that live there already, these parasites here. And so to colonize this niche, the metastatic cells need to actually activate a mechanotransduction pathway that allows them to spread and migrate over the abluminal surface of blood vessels, physically displacing the parasites as they go. And so the Massaguet lab had studied the molecular requirements for this pathway, and they had identified a number of proteins, including some cell surface molecules, some signaling proteins, but also these mechanosensitive transcription factors, YAP and MRTF. And what they had found is that depletion of either one of these proteins actually inhibits this response. But they had been doing all of their experiments in immunodeficient mouse models. And so they started doing some experiments in immunocompetent models and getting some strange results, which sort of got us interested. And we were particularly interested in MRTF for reasons that I'll describe in a bit. And by we, I mean Maria Teo, a postdoc in my lab, and Emra Ura, who was then a postdoc in the Massaguet lab. And so before I get into why we were so interested in MRTF, I want to tell you a little bit about what MRTF is. It's a transcription factor, a family of transcription factors. It's actually regulated by monomeric actin. So if there's monomeric actin present in the cytoplasm, it binds to MRTF and it sequesters it there. However, if that monomeric actin is consumed into actin filaments, the MRTF then goes into the nucleus where it drives the production of more actin and actin binding proteins to essentially fuel this architectural response, this spreading and migration response, right? And so We became interested in this project based on the following experiments. So these were just standard metastasis assay experiments. And so what we took were B16 melanoma cells and inject them IV into mice. They colonize the lung and you monitor that colonization. So what we found was that when you deplete MRTF, you inhibit this colonization response. And this was not surprising at all. This is because MRTF is required for the structural response that allows for the invasion. What was more surprising is that when we overexpressed MRTF, we found that it also inhibited the colonization response. And the most interesting thing about this is that when we took these mice and depleted NK cells before we injected the cancer cells, we actually got the result that we initially expected, and that there was more colonization by the overexpressing cells. So the capacity of MRTF overexpression to inhibit colonization depended on cytotoxic lymphocytes. And so the way we sort of conceptualized this was the following, is that MRTF activity actually promotes metastasis in the absence of cytotoxic lymphocytes. But when there are cytotoxic lymphocytes present, MRTF activity exposes this immune vulnerability. And so you get this sort of Goldilocks response, right? And we were really interested in this because of MRTF's established role in driving structural transitions in cells. And so we were wondering whether this immune vulnerability might have an architectural basis rather than sort of a standard molecular basis. And so we did some functional experiments first, and these are just in vitro assays showing that cancer cell lines, these are melanoma breast cancer cells, overexpressing MRTF are actually more sensitive to killing by cytotoxic T cells. And not only that, they actually just stimulate stronger T cell activation. So this is looking at degranulation by cell surface exposure of LAMP1. You see that the cell lines that are overexpressing MRTF drive stronger responses. So could this have an architectural basis? So we looked at the cells, and what we realized was that the MRTF overexpressing cells look very different in terms of their cytoskeletal architecture. They have these copious stress fibers that are sort of supporting their architecture, suggesting that they might actually feel physically different. And so to get at that, we performed atomic force microscopy, which is this single cell biophysical approach where you bring a cantilever down on the surface of the cell and then monitor its displacement by bouncing a laser off the back of the cantilever, so you can measure stiffness. And so these are some representative data. Each of these is a whole cell. Each of these pixels is one pushing event, and they're color-coded for stiffness. And so you can see that the MRTF-A and MRTF-B overexpressing cells are stiffer than the control cells, and this is just quantification below. And so everything that I've showed you thus far is consistent with this idea that if you make a cell stiffer, you sensitize it to cytotoxic lymphocytes. But the problem with doing mechanobiology in the 2020s is that we've basically given biochemistry, molecular biology a 30-year head start. And so if you want to convince anyone that something has a mechanical basis, you kind of have to rule out 30 years worth of biochemical hypotheses. And so the obvious biochemical hypothesis here was that, well, you're just overexpressing a transcription factor, so you're inducing the upregulation of a co-stimulatory ligand that is driving enhanced activation of the T cell or the NK cell biochemically. And so we started going through these candidates one by one, and we soon became exhausted, and because there are thousands of these molecules. And so we realized that what we had to do was find some way of experimentally decoupling what was happening on the surface of the cell from what was happening on the cytoskeleton underneath the surface. And so we did this a couple different ways. I'll just tell you about one of them. So this is a bacterial protein. It's called a DACT by the people that characterized it. But what it is it basically ADP ribosylates actin, and this melts actin fibers in cells. And so just to give you an example, here DACT has been linked to RFP. So you'll see all the red cells in which DACT is expressed, they have little to no actin. And this induces a precipitous decrease in their stiffness, right? So we could do the following experiment where we could take our cancer cells, either regular cancer cells or cancer cells that were overexpressing MRTF, and we could either transiently transfect them with empty vector or with this DACT vector to depolymerize the cytoskeleton. And then we could co-culture the cells with the CTLs to look at activation. And so the reason we use DACT, of course, is that we want the CTLs to have their actin, right? We want the actin in the C cells to be intact. And so this is the result I showed you earlier. If you just use to make cancer cells more stimulatory to T cells depends on the cytoskeleton and not on the cell surface, right? And so based on these results, we developed the following working model. So when metastatic cells invade new organs, they activate an MRTF dependent mechanotransduction pathway that allows them to spread and migrate and invade. This enhances their invasive capacity, but it
Recording in
stimulates
progress.
the destruction of the cancer cell. And so we called this process mechanosurveillance because it's fun to coin new words to describe biological phenomena. And we're very interested in examining the different contexts in which mechanosurveillance, say in senescence or in the clearance and identification of virally infected cells. But what I'd like to talk about for the rest of the seminar today is a new direction we've taken this, where we've looked at the how mechanosurveillance depends on the microenvironment. So this initial work that we did on cancer cell stiffness, it really hinged on a cell intrinsic regulator of stiffness, which is MRTF. But cell stiffness can also be regulated cell extrinsically by the microenvironment. And this is a process called mechanoreciprocity. So it turns out that adherent cells grown in stiffer environments become stiffer themselves, and adherent cells in softer environments are softer. They tend to mimic the mechanical properties of their environment, right? And this this concept of mechanoreciprocity is very interesting in the context of metastasis, because as all of you know, metastasis takes place in a wide range of target organs that vary in their mechanical properties dramatically. So breast cancer famously metastasizes to three main organs: the brain and the lungs, which are very soft, and the bone, which is the stiffest material in metazoan organisms. And so if mechanoreciprocity holds, one might expect that the cancer cells colonizing the bone would be stiffer than the cancer cells colonizing these other organs. And if mechanosurveillance holds, one might expect then that cytotoxic lymphocytes would be particularly effective or important at policing and destroying cancer cells that were colonizing the bone. And what that would imply is that mechanosurveillance would not only control how much metastasis takes place, but also where it grows, the metastatic site selection, right? And so we became very interested in this idea. And by we, I mean Yasmin Obana, who's a very talented graduate student in the lab. And so I'm just going to show you some in vitro validation of these concepts. And so what we did here was we grew cancer cells on substrates of different rigidity. So basically soft hydrogel and stiff hydrogel substrates. And then we detached the cancer cells after an overnight culturing, and then performed AFM on them. This atomic force microscopy. And what we found was that the cells on the stiffer substrate tended to be stiffer than the cells on the softer substrate. But you'll notice that these results are not particularly impressive and not particularly statistically significant. And this is because AFM is a sort of a low throughput process. And so what we've been taking advantage of is new methodology developed by Scott Manalis, our collaborator at MIT, and Ye Zhang in his lab. It's called the suspended microchannel resonator. And what this is really a hairpin microchannel that you push cells through. And the channel is vibrating continuously. And it turns out that if the cell is in the node of this And so this allows us to do much more robust experiments. So you'll see here a lot of different substrate stiffnesses were assessed. and a lot more cells were analyzed. And so we can isolate statistically robust data, and you can see here that cells cultured on surfaces of increasing stiffness become stiffer. So this is basically in vitro mechanoreciprocity. So what about mechanosurveillance? So we did the same sort of experiment. We grew cancer cells on substrates of increasing stiffness, and then we added in ovapeptide and cytotoxic T cells, OT1 T cells that were responsive to ova. And what we found was that this is looking at NK cell mediated killing in the same type of context, right? And so next, what we wanted to do is to assess whether this sort of reciprocity and surveillance were actually taking place in vivo. And so I realized that in the context of metastatic site preference, our hypothesis made two predictions. The first is that bone metastasis would be particularly sensitive to cytotoxic lymphocytes in vivo. And second, is that metastatic cell stiffness, the actual stiffness of metastatic cells should reflect the effects of mechanoreciprocity and mechanosurveillance in vivo. And I'll get to what I mean by this in a bit, but we'll start with the first prediction. So we went back to our metastasis model, injecting melanoma cells IV into mice. And what we did was we injected wild type mice, immunocompetent mice, and also mice that were lacking in cytotoxic lymphocyte function, actually NK depleted mice or perforin knockout mice. And what we found was not only did we see more metastasis in these mice, but we also started seeing metastasis here in the long bones of the leg. These are the femurs, right? And you might say, well, you're getting more metastasis, so you're getting more metastasis everywhere. But if you take wild type mice and inject 8 times more cancer cells, you get great metastasis formation in the lungs, but not in the legs. And so this is just quantified here. Looking at total metastasis, you can actually rescue total metastasis in control recipients with just a large number of cells. However, it doesn't change the proportion of metastases in the bone, which we've measured here by looking at the ratio of legs to total metastasis. That requires a cytotoxic lymphocyte deficiency. These are what the bone metastases look like. And so this is a femur, the end of a femur. So what the cancer cells like to do is they like to live in this region here, this metaphyseal region, where there's a lot of trabecular bone. There are these sheets of bone inside of the marrow space, and the cancer cells like to grow along these sheets, and then they sort of push into the shaft of the bone. We also see metastatic colonization of the spine in these mice. So this is a cross section of the spine. They like to live in the vertebral body here, which also contains a lot of trabecular bone. But then the tumors will pop out into this epidural space and start impinging on the spinal cord. And we should have known that this was happening because you can actually read out this phenotype behaviorally. So with perforin knockout mice that are injected with B16 cells, you actually end up having to sack them because they start dragging their hind legs, indicative of hind leg paralysis. And you can even read this out epidemiologically. So perforin knockout mice, they don't die any faster when you put in B16 cells, but they just die, or you have to sack them because of paralysis rather than sort of lung distress. We see the same sort of pattern with this NK cell depletion model. This isn't just true for B16 melanoma. We're doing the same sorts of experiments with EMT6 breast cancer cells, and as with the melanoma, we see that when we deplete CD8 T cells, in this case, we see an expansion of bone colonization. Okay, so that addresses the first question. Cytotoxic lymphocytes appear to be particularly important for policing bone metastasis. The second prediction is that the mechanical properties of the actual metastatic cells would reflect the effects of mechanoreciprocity and mechanosurveillance. And so to get at that, we leveraged this suspended microchannel resonator approach. And what we did was we extracted metastatic cells from the lungs of control mice, and from the lungs and the bones of perforin knockout mice. We took the metastases out, we dissociated them, we purified the cancer cells, and then we put them over this stiffness sensing device. And these are the results of those experiments. So the cancer cells that were taken out of the bone are stiffer than the lung samples, and this is indicative of mechanoreciprocity. So they're a stiffer microenvironment, they respond by stiffening. But you'll notice that if even comparing the lung metastases from perforin knockout mice and wild-type mice, the perforin knockout derived metastatic cells are stiffer than the wild-type derived metastatic cells. In particular, there's this population here that appears to disappear in the wild-type, and this is indicative of mechanosurveillance. So these stiffer cells are selectively destroyed by cytotoxic lymphocytes, and so we see the same pattern of results in the NK depletion model. So the bones and the lungs they differ mechanically, but they also differ in pretty much every other property you can think about. And so what we wanted to address was whether environmental stiffness could actually dictate this mechanoregulation as an independent variable. And so to get at that, we've been working with Jungwoo Lee, who's a bioengineer at UMass Amherst, and the grad student in his lab, Jungwoo Kwok. And what the Lee lab has developed are these hydrogel growth niches. They're like little hydrogel hockey pucks, and you can implant these subcutaneously into mice, and over a 2 or 3 week period, they become vascularized. And then when you inject cancer cells into the mice, the cancer cells will actually occupy these niches, or colonize these niches. And so what we did was we took a stiff hydrogel niche and a soft hydrogel niche. We implanted them into mice, and so we sort of paired them, and then we either depleted the mice of NK cells, or we left them the NK cells intact, and then we injected the cancer cells, and then measured the relative colonization of the stiff and soft hydrogels in each mouse. And this is just to show you what one of the mice looks like after surgery. These are these subcutaneously implanted niches. This is what one of the niches looks like from the opposite side after vascularization. And these are the results of the experiment. So in the NK depleted mice, we consistently saw better colonization of the stiff hydrogel than the soft hydrogel. And this difference went away in the control mice. So you bring back cytotoxic lymphocytes, and all of a sudden you don't see a difference anymore. And so what these results are, they're consistent with the idea that you actually get the cancer cells prefer to colonize these stiff niches, but the NK cells prefer to kill cancer cells in these stiff niches. So indicative of mechanosurveillance. Okay, so we wanted to get a sense of the molecular determinants that were driving this process. And so, like every other immunology lab these days, we turn to single-cell RNA-seq. And so we took the same 3 samples, metastatic cells from wild-type lung, and metastatic cells from perforin knockout lung and perforin knockout bone, and we did comparative RNA-seq. This is just a UMAP of just the cancer cells. As you can see, there's some minor differences moving from wild-type lung to perforin knockout lung, and some much more dramatic differences when you look at the cancer cells in perforin knockout bone. There's a major shift in population distribution. In particular, there's this cluster 7 that is dramatically enriched in perforin knockout bone. And so we did the differential gene expression analysis of markers that characterize this population. And one gene stood out to us in particular, which is called osteopontin. It really characterizes this population. And we also found that osteopontin was upregulated in bone metastases in this NK depletion model as well. So what is osteopontin? It's a secreted molecule that's very characteristic of cells in the bone microenvironment. It incorporates into the extracellular matrix. It has a number of cognate receptors, including CD44 and certain integrin proteins. It also has some characterized immunomodulatory activity. And in addition, there are some papers that suggest that there's an intracellular form of osteopontin as well as in extracellular form. We were particularly interested in this molecule because it's highly expressed by bone resident cells, osteoclasts and osteoblasts, and also that it's been shown to mediate osteoclast attachment to the bone matrix. And so we were curious as to whether it might be doing the same, allowing cancer cells to recognize bone, to adapt to it, and to move in. A sort of interesting tidbit: if you look at osteopontin in the TCGA database, you can divide you can look at cancers where osteopontin is deleted or downregulated, and then you can look at cancers where it is amplified. And if you order the cancers by their stiffness or the stiffness of the parent tissue, one of the things you notice is that the osteopontin amplification is more prevalent in stiffer cancers, and whereas osteopontin deletion is more prevalent in softer cancers. And so that was intriguing to us as well, because it implied a role in the mechanoregulation of these processes. And so we started taking a look at osteopontin in cells. This is just a B16 cell sitting on a surface in vitro, and what we found is that osteopontin tends to accumulate in these bundles that overlap actin bundles inside the cell. Importantly, it's unclear whether this osteopontin staining is actually outside the cell or inside the cell. Well, we went ahead and made some knockout cell lines. And this is just a Western blot validating the quality of the knockout. As you can see, we need to actually treat the cells with Golgi block in order to see the osteopontin. And what that indicates to us is that the vast majority of this protein is actually being secreted. What we noticed is that there were some architectural changes in these knockout cells. So this is another staining experiment looking at actin and osteopontin. And not only is the osteopontin signal diminished in the knockout, but the actin signal is also diminished. And that's shown here. That's suggesting that osteopontin is important for maintaining the mechanics of these cells. And that indeed is the case. So this is some stiffness analysis of osteopontin knockout cells, and the knockout cells are indeed softer than the wild type cells, and so it's important for maintaining stiffness. We think that this is specific to osteopontin because we can actually take osteopontin, purified osteopontin protein, and add it back to these knockout cells, and it increases their stiffness. This is after an overnight incubation in the osteopontin protein. It also controls their stimulatory capacity when we look at cytotoxic lymphocytes. So here we're looking at wild type target cells versus osteopontin knockout cells. The knockout cells are less effective at being killed by CCLs, less effective at stimulating degranulation and cytokine production. So what I've shown you thus far indicates that osteopontin controls the mechanical stiffness of cancer cells. But what about the ability of these cancer cells to actually respond to environmental stiffness? And so to get at that, we went back to this experiment where we grow cancer cells overnight on surfaces of increasing stiffness. And then we see whether the cancer cells stiffen or they undergo mechanoreciprocity in response to these surfaces. And so we compared wild type and knockout cancer cells. And this is the results I showed you earlier. So if you grow wild type cells on surfaces of increasing stiffness, the cells stiffen. Interestingly, when you take osteopontin away, you completely abrogate this mechanoreciprocal response. And so the cells can no longer respond to environmental stiffness. So what are the implications of this in vivo? And so this is kind of a complicated experiment because we're taking wild type and knockout cells and we're putting them into wild type and perforin knockout hosts. But I'll go through it step by step. And so then we're looking at colonization of the lungs and the legs. And so if you look over here, these are wild type cells into wild type and perforin knockout hosts. This is the results I showed you earlier. When you take cytotoxic lymphocyte activity away, the wild type cells, they selectively expand in leg metastasis, right? So this preference for leg metastasis completely disappears when you take osteopontin away. In fact, we have trouble documenting any bone metastasis in osteopontin knockout mice. And so osteopontin appears to be required, and osteopontin-dependent mechanorecipropathy specifically appears to be required for bone colonization during metastasis. So this addresses mechanorecipropathy. What about the role of osteopontin in mechanosurveillance? Okay, so to do this, we took advantage of the fact that the stiffness properties of metastatic cells that are removed from perforin knockout lungs are different from the stiffness properties of metastatic cells in the lungs of wild type mice. So we basically injected a wild type or osteopontin knockout cells into wild type perforin knockout recipients, but then we looked at the lung cells by SMR, the stiffness flow cytometry. And so this is the result I showed you earlier. With wild type cancer cells, the metastatic cells that you extract from the lungs of perforin knockout mice are stiffer, and the wild types, the cells from wild type mice are softer. And this sort of selective elimination of the stiff population is indicative of mechanosurveillance. This difference is not apparent when you use knockout cells. So to the extent that osteopontin knockout cells are being targeted by immune cells, they are not being targeted on the basis of their stiffness. And so based on that, we've developed the following working model that deals with the interplay between mechanorecipropathy, stiffness sensing of the environment, and mechanosurveillance. And so basically, mechanorecipropathy acts like a dial to essentially control the strength of mechanosurveillance. So in stiff organs like the lung, cytotoxic lymphocyte dependent mechanosurveillance matters a lot more, and so it has much more of an effect. So you get a selective destruction of cancer cells in the bone. It's more effective there. And then when you take cytotoxic lymphocytes away, you get a selective expansion in the bone. This is our current model. What I'd like to spend the last few minutes discussing is just some attempts that we've made to interrogate these concepts in human patients. And so there hasn't been a lot of analysis of mechanosurveillance in human patients. What we did initially was look through published single-cell RNA-seq data sets to identify kind of a good context for addressing this hypothesis. And we found this really interesting study where they took breast cancer patients and treated them with anti-PD-1, but it was a very controlled study in the sense that they took samples right before a single dose of anti-PD-1 and then 7 to 9 days after. And then they evaluated these samples based on whether they observed clonal expansion of T cells in the tumor microenvironment, right? So they were looking directly at T cell activation. And so those were classified as responders versus non-responders. This is a UMAP of the entire data set. I think there were 40 patients in total. The responders are in green, the non-responders are in brown. But what we noticed when we looked at the gene expression of responders versus non-responders is that the responders, there was this distinct upregulation of molecules that were associated with the actin cytoskeleton, specifically the actin cytoskeleton, suggesting that this response to checkpoint blockade therapy might be driven in part by the stiffness of the cancer cells. If you take a closer look at this UMAP, and here it's colored for expression of actin cytoskeletal related genes. This is beta actin. This is a molecule called profilin. Here we compared this to beta-2 microglobulin, which is, of course, important for class 1 expression and presumably for immune targeting. What you'll notice with the beta-2 microglobulin is that the responder populations tend to express more beta-2 microglobulin. This is consistent with responder populations being more likely to elicit cytotoxic lymphocyte activation, right? But you'll notice that there's some variation in the expression of beta-2 microglobulin among responder subsets. And what was sort of interesting is that we noticed that in some of the responder subsets that had relatively lower expression of beta-2 microglobulin, you actually see higher expression of these actin related genes. And what that indicated to us is that perhaps increasing stiffness could actually compensate for suboptimal beta-2 microglobulin expression in eliciting a T-cell based anti-tumor response. So this is sort of intriguing links potentially to mechanosurveillance, but we wanted to actually look at these phenomena in human metastasis. And we wanted to really address the role of mechanoreciprocity and mechanosurveillance in human metastasis as we had done so with mice. And so the problem we faced really was that we couldn't just start comparing random bone metastases and lung metastases and brain metastases, liver metastases from different patients, because the differences that we observed, the biophysical differences, the gene expression differences that we observed would presumably be dominated by the underlying genetics of each patient tumor and the patients themselves, right? And so we ended up addressing this through this really nice collaboration that we started with Ori Barzilai, who's a surgeon at MSK. And what Ori specializes in are tumors, metastatic tumors that grow in the spine. And a number of the tumors that he excises have this interesting property where a part of the tumor is in the bony part of the spine, and then a part of the tumor pops out and is in this epidural space around the spinal cord. And so and the mechanical properties of these two environments are very different. So this is, you have access to very stiff bone here, and this is mostly just very soft connective tissue and fat. And so what you have is a physically contiguous tumor that's occupying two mechanically distinct locations. And so what Ori does is he can give us a piece of this part of the tumor and a piece of this part of the tumor, and then we can run our comparisons. And so this is just an example of one of these tumors. You see it constricting the spinal cord here. And so thus far, we've focused on non-small cell lung cancer and breast cancer, and we've done two of each. And so I'll show you what we've got so far. And so what we did initially was we isolated the cancer cells, we purified them by using the EpCAM marker, and then we performed SMR, that stiffness flow cytometry, and so these are the two breast cancer samples, and as you'll see, the cancer cells extracted from the bony part, or the osseous part of these tumors, are quite a bit stiffer than the cancer cells extracted from the epidural part. And this is totally consistent with the idea of mechanoreciprocal stiffening in the bone microenvironment. So we were really excited when we saw these results. We were actually much less excited when we saw the lung cancer results, which showed no difference in stiffness at all between the samples. And so that sort of threw us for a loop. But then it occurred to us that what these differences might reflect are the relative strength of the anti-tumor immune response in these two different tumor types. So breast cancer is considered to be quite a bit immunologically colder than lung cancer, and we validated that by doing some staining for T cells and NK cells, and we found that there was very little lymphocyte in the breast cancer cells. But much more prevalent lymphocyte infiltration in the lung cancer samples, and this is sort of consistent with prior literature. And so the idea here is that this lymphocyte infiltration might leads to the selective elimination of the stiffer cells that would have accumulated through mechanoreciprocity in this osseous environment. And so that's a diagram here. So in the case of breast cancer, you have mechanoreciprocity in the bone, but in the face of low immune pressure, that actually manifests in a stiffness differential. Whereas with lung cancer, the mechanoreciprocity that would take place in the bone in the face of high immune pressure results in the selective elimination of these stiffened cells and no observed stiffness differential when we end up taking out this cells. And so we're hard at work trying to generate more of these samples and to extend this analysis at the moment. But for today, that's really all I have to say. I just want to thank again the people that did the work. Maria Teo really drove the first part of the talk on MRTF, and the work on metastatic site preference has been really spearheaded by Yasmin Elbana. We've had a ton of really critical collaborators on this project from a bunch of different places, and I want to particularly acknowledge the people highlighted in cyan here, as well as the patients. Thanks so much for your attention.
Recording stopped.
Well, thank you so much, Morgan. That's absolutely refreshing. Really great, comprehensive illustration of the mechanical immunology. We can start the question session. If anyone have a question, please raise your hand or put your question in the chat box. Xiaolei, go ahead.
Thanks, Tran. Hey, Morgan. Nice to meet you. This is such a fabulous work, as always. I think I have two questions. One is about the MRTF. So it looks like it provides a positive feedback because whenever there is a soluble acting, it inhibits its transcriptional activity. So I wonder, does the expression level of the MRTF affect its metastatic site? In other words, how heterogeneous the expression level of MRTF is in the cancer cells?
Xiaolei, it's really good to see you. We have looked at MRTF expression what we didn't look at MRTF expression, or we've stained for MRTF in tumors, and what we found is that not only is there heterogeneity in MRTF expression, but where you really see a lot of heterogeneity is in nuclear localization of MRTF. What we found, and this was in Maria's paper, is that when cytotoxic lymphocytes are present, when there's cytotoxic lymphocyte pressure, you actually see fewer cells with nuclearly localized MRTF. Suggesting that it's being selected. Your first question, we have not looked at whether MRTF overexpression affects metastatic site selection, but one would predict that those cells would not be particularly good at colonizing the bone if cytotoxic lymphocytes are present. So you would then amplify this response that we're seeing when you take cytotoxic lymphocytes away, you get particularly good colonization, right? But yeah, we haven't done that experiment.
Okay, that's great. I think a related question is, I think it is from the therapeutic perspective, is any of these mechanosensing pathway is druggable or?
Yeah, so I mean that's a really good question, and obviously something that we're looking into. Initially, when we started on this work, it didn't seem too promising because you can't really modulate actin and myosin and expect to have a selective effect on tumors. Although some people have tried, some people are trying. So the more we look into this, the more we identify molecules like osteopontin that may be a little more selective for specific microenvironments. The thing about osteopontin, though, is its role is complex, right? So one might imagine that if it were important for bone colonization, just in the initiation of bone colonization, you actually want to block osteopontin. But then after metastasis is established, you might actually want to enhance expression because it would apparently sensitize. These sorts of, I think these are very complex considerations, the therapies will still need to be very, very much tailored to match the state of disease and whatnot. I mean, one of the things that I've always been intrigued by, but not quite sure how to address, is that we always talking about using chemical tools to address these sort of physical phenomena, right? But to the extent that there is a mechanical dimension of these interactions that are important, it may be interesting thinking about physical modalities that could be used. And I'm not saying I know how to do this, but things like ultrasound and MRI. The nice thing about that is they can be targeted as well, right? And so anyway, that's just a random thought.
Well, absolutely. I think this like mechano it's a completely like new dimension to think about the cancer immune surveillance and the cancer therapy. I think that's truly exciting. Thank you. Thank you so much, Morgan, and nice to meet you.
It was great to see you. Great to
Yeah.
see
you. Actually, I have, before Paul's question, I have 2 questions for the first part of the talk. So actually, I'm curious about this MRTF regulation machinery, because I mean, it seems to me like during your talk that it's mainly or dominantly function during the metastasis cancer cell or cancer cell itself. So my first question is how it is regulated, the MRTF. I mean, it's only being regulated in the cancer cells or during the metastasis. How it's actually, what's the factor upstream of that?
So there are various ways to turn MRTF on or off, but it's regulated also at the level of nuclear localization, and so that's part of the process that's driven by actin, right? And so as Shelley was saying, MRTF activity is really kind of a feedback sort of relationship, at least as traditionally conceived. So the cytoskeleton, so the cell decides it wants to expand its cytoskeletal footprint. MRTF responds to a deficiency in building blocks, and so it will drive that up. And so what we're doing when we're overexpressing MRTF is we're really just kind of resetting that set point. So when you put more MRTF into the cytoplasm, you essentially and there's no actin there to bind it, it goes into the nucleus. And so basically you have to make a cell. You have to generate a lot of excess actin in order to stop that process at that time, or more excess actin to stop the process at the moment. I don't know very much about transcriptional regulation of the MRTF gene itself, but this feedback with its own sort of structural elaboration is something that is responsive not only to what the cell does, but also to the environment, because the environment will like induce adhesion formation, which will drive more filament production itself. I'm not sure I understand. Did I answer your question at all?
Yeah, I mean, first of all, the transcription regulation, I mean, like you said, probably is still unknown, but the other thing is the MRTF probably is, as it's a protein, it's also have its own property for its own activity. But I just also wondering that its activity or the binding capacity will be somehow different during the cancer development in the cancer cells.
So its capacity for binding actin. I don't know if that's been shown. But another thing is that it interacts. It regulates transcription as a complex with the serum response factor. So that's another level of regulation that one could imagine is titrating away binding partners and things like that.
I see. My second question actually is, I mean, this is actually completely new for me. So one thing I'm actually curious is that the stiffness of the cell is only occurring on the cancer cells, or it's reciprocally regulate between the cancer cell and partner of the lymphocytes. Whether that during the lymphocytes interacting with the cancer cells, it's also sort of influence the stiffness of the lymphocytes, which potentially lead to lymphocytes change its function.
Yeah, that's a really interesting question, and we have not done that much to address it. We did do a study that was published last year where where we stimulated cytotoxic T cells with stimulatory hydrogel beads of different stiffness.
Right.
And then we looked at the deformation of patterns of the beads, or the degree of deformation that was induced by the T cells. But what we found is that when we changed the stiffness of the bead that we gave to the T cell, not only did we change the amount of deformation, and I mean that's sort of simple because or trivial in a certain sense because it's stiffer, so you're not going to be able to deform it much. But it seemed like if we looked at the actin pattern of the interface, that the T cell was actually changing its mechanical strategy. So so with softer particles, it was more of a situation where it was creating a lot of protrusions to push into the particle. Whereas with stiffer particles, it kind of peripheralized the structure. So more of the actin was accumulating at the periphery with a sort of a gap of actin in the middle. So the lymphocytes are definitely responding to stiffness differentials or mechanical differences in their target.
But the experiment you're just describing is still sort of an extrinsic impact. But when you actually describe the stiffness from the cancer cell perspective, it's more like you're actually talking about intrinsic, for example, MRTF. So I'm also wondering whether that, when you have this interaction between the cancer cell and the lymphocyte, whether any intrinsic things will happen, intrinsic
to
machine
the
will
lymphocyte that will,
yes, to the
that will
lymphocyte.
change its
Right.
mechanoregulation. Yeah.
Yeah.
I mean, no, that's a really interesting question. We've not looked at that.
Because when you think about this, it's also have all these immune synapse, which one part of your research program. And then this immune synapse formation, I guess it's also have something to do with the lymphocytes and tumor cell stiffness. Because, one hand, cancer can change their property, but at the same time, if you want that reciprocal interaction from the lymphocytes, somehow the mechanical property from the lymphocytes should be different as well in order to cope with this situation.
So I think there are things that happen on a short time scale, right? Which are like what I described, right? But what you're talking about is a mechanical interaction that changes really the cell state, the transcriptional state of the cell, and so that there are not a lot is known about that, but there are indications that is quite important. I think the context in which that's been studied is the differentiation of naive cells into effectors. And so there's a whole field where people will do that under different mechanical situations. So they basically will make different materials to stimulate the cells, and then look at the quality of the effector cells that grow out, right? And what they've shown is that the stiffness, lateral diffusibility, et cetera, of the stimulatory surface, it actually makes a big difference in the quality of the cells that you get out. And so the capacity to make structures and to exert force would definitely be part of that, I think. And so I'm sure what you're saying is true. I just don't think there's a lot that's known about it yet.
I see. Okay.
Yeah.
Paul, you there?
Hi, Morgan. Yeah, very nice, interesting talk.
Thanks.
I really like it. So very smart angle. So two questions. I probably will from a big question first. So in your last part of your talk, like metastasis to the bone, it can include different cancers like breast cancer, as you mentioned, maybe lung, right? So how bone environment regulates stiffness of cells differently, and then are they correlated with the molecule like OPN? Is that no?
So, I mean, there's a lot of interesting questions there. So the way we imagine the bone environment, or the bone matrix itself, dictates stiffness is through adhesive contacts that were sort of analogous to what we see in vitro when a cell spreads on stiff plastic or something like that. Whether the role of OPN is really interesting. So there have been there's been some immunohistochemistry that indicates that OPN kind of accumulates at the interface between cells that are in the marrow space and the bone matrix itself. It also is known to sort of stick to mineralized bone, and so the idea is that I think the idea we favor is it's acting as kind of like an adhesion adapter. It sort of enables the cell to grip bone. A really interesting question is osteoclasts, a number of bone measurement cells, they make a lot of osteopontin. So why is it so important for the cancer cell to make osteopontin in order to move in? And that's something that we really don't know. But it seems that the osteopontin that is there already is somehow being used, or bound up or occupied or whatnot. So the cancer cells need to generate their own pool of osteopontin. Another really interesting question about the bone microenvironment is that, which I sort of conveniently ignored, is that sure, yeah, the bone's really stiff, but the marrow is incredibly soft. And so I've been talking about, cancer cells growing on the bone, but what about the cancer cells that pop off into this space in the middle? That's a very different mechanical environment. And so what you would expect is not only mechanical adjustment to the bone, mechanical adaptation to the bone, but quite a bit of mechanical diversification within the tumor itself. And one of the things that we've noticed about these metastatic populations that we extract from the bone is that when we look at their stiffness, there's a huge range in stiffness. And so we're gearing up now to do some spatial transcriptomics to sort of see if we can map that anticipated heterogeneity within the bone microenvironment.
Okay, so another question that I have is that, so you're talking about a lot of cytoskeleton that will affect the stiffness, right? But maybe I'm too naive, but there's maybe some other ways to enhance the stiffness, right? For example, the structure of the membrane, like, sphingomyelin, cholesterol, or, when cell become hypertonic, right? Like swollen.
Yeah.
It can be also different too, but like you did not mention that part. I'm just curious about what you think.
I have not, but I think you're right. It absolutely is a parameter that's important, and in fact, Li Tang at Lausanne has shown that changing the cholesterol content of cancer cell membranes can affect their sensitivity to cytotoxic lymphocytes. And so these are all, and we had a study last year where we showed that in a kind of a roundabout way, that enriching sphingomyelin and ceramides in neutrophil membranes could change their membrane tension and their functional capacity. And so the membrane composition is also critical. So
how about like to make the cell like swollen, right? Like, for example, bone can swollen cell differently, like a lung versus like a breast cancer, like they have different regulation, right? So then would that be more sensitive to one of the cell can be more sensitive to the kill?
In fact, we showed a number of years ago that if you hypotonically swell a cancer cell, you can make it more sensitive to perforin and granzyme-mediated killing. The thing about swelling, though, like acute swelling, is that cells are very they seem to care a lot about their membrane tension, and so if you hypotonically stress a cell,
right,
its membrane tension will increase for about 25 minutes, and then it'll adjust. And that's and so from a therapeutic standpoint, that might be harder, because the cells won't maintain that change in
How about temperature? If you lower the temperature,
or if you lower the temperature,
right?
Oh,
to make it more rigid, right? Because it's not like a fluid anymore.
I don't know. It would seem that you might have problems, sort of with your T cells at that point, or with your lymphocytes.
Yeah. All right. Yeah. Thanks so much. Yeah. Very interesting.
No problem. Thank you.
Jack, are you here?
Yeah.
Go ahead.
Hey Morgan, great to see you
Hey,
again.
how are you?
Hey,
It's
long
good to see
time
you.
not
Yeah,
seeing you.
yeah.
Yeah. So actually, I always have some question regarding this stiffness because you are looking at this cellular stiffness. So, but in the tumor microenvironment, the actual cellular matrix also change, right?
Mm, mm.
And they also change the microenvironment stiffness. So how these two stiffness differentiate regulate this, like for example, T cell killing, which one is more important?
Mm. So I think environmental stiffness will affect the stiffness of cancer cells, right? And then that will sort of sensitize them mechanically to cytotoxic lymphocytes. That being said, I think particularly the deposition of extracellular matrix can have a bunch of other effects as well. And this has been studied, I think, extensively in the context of primary tumors, where it's been suggested that they could actually physically block the ingress of immune cells or promote like cancer cell EMT and things like that.
Yeah,
Um,
yeah,
and all of those, I think all of those phenomena are occurring. And so the problem with studying those in the context of like a very complicated primary tumor is that they're all sort of overlaid on top of each other, right? And so that's part of the reason we've been focusing on metastasis in these studies of mechanosurveillance is that we wanted to try to find a situation where it would be as close as we could get to, one cytotoxic lymphocyte, one cancer cell in an environment. And of course, I showed you all those pictures of those tumors that had grown out. But if you do tumor experiments, you're always like evaluating what happened um, um, 3 or 4 weeks after the really important stuff happened, right? And so you're sort
yeah.
of inferring what happened before that. And so the situation that we're trying to create is one where we've simplified things so that we can isolate the effects of the mechanical environment on like one cancer cell and its sensitivity to immune cells. Once the tumor gets large enough to create its own immune microenvironment or to
Yeah.
create its own immunosuppressive microenvironment, then yeah, I mean, you have a bunch of sort of mechanisms that are working kind of at cross purposes. I'm not sure if did that really address your question, or was that?
But in my mind, usually in the single cell level, these metastatic cells, tumor cell, usually like softer, right? I
Yeah,
remember,
they are.
right?
Yeah,
So
yeah.
T cell seems harder to kill there compared to these other cells, okay? But ECM usually it becomes stiffer, okay, during the tumor growth, okay? So I don't know which one. That's why I'm asking which
Yeah.
one is more, important for regulating T cell killing, for example, right?
So we sort of like to think of it in terms of vulnerabilities, right?
Yeah,
It's been shown that tumor cells are softer. More
yeah,
malignant cells are softer than their sort of like their normal cells of origin. Um, However, in order to I guess if we wanted to speculate as to why this sort of mechanosurveillance evolved, it's that tumor cells are softer, but in order to push into a new organ, they kind of have to stiffen. They have to stiffen transiently in order to like create space in this perivascular niche, right? In the case of
okay,
the MRTF study. And so there's no way around it. It's unavoidable. And so these unavoidable steps that tumor cells have to take, they're generally the sources of vulnerabilities that can be targeted by the immune system or therapeutically or
I
whatnot.
see,
And
I
we think
see.
that we sort of feel the same way about colonization of the bone, is that in order to make those initial steps to colonization, they have to bind to the bone matrix and they have to stick to it and they have to physically respond to it. And so the immune system can take advantage of that.
Right,
To
right, right.
recognize this kind of this
Yeah, I
dysregulation.
see what you mean. So,
Um,
I actually two more short question. Okay,
sure,
the
yeah.
second one is so last year there's a paper from Professor Zhou's in China, and it shows the stiffness can promote T cell exhaustion. Okay,
Mm,
right.
mm.
You probably read that paper, and your data shows the stiffness can promote the T cell killing. of tumor cells. So which one is more important? Okay, one is T cell exhaustion, right? One is boosting the T cell killing. So in vivo, which one is, I think maybe there's some critical balancing point for T cell that can keep the longer, killing property.
Exhaustion is really just a manifestation of good activation that sort of takes place over over an extended period of time. So,
right.
So there's always this sort of there's always this trade-off between exhaustion and activation. You need activation in order to get, but the key thing for T cells, because they seem to have evolved to respond vigorously to something for a short period of time, right?
Yes.
And so I guess from a therapeutic standpoint, you want to try to set up situations where you expose T cells to something with lots of ligands, something that's super stiff, something for like a short period of time, and then you let them rest. And then I'm not sure how to do that, but that's a way to sort of conceptualize both these exhaustion results and the results that we have under the same umbrella. It's like you get stronger activation with stiffness, and that's totally consistent with enhanced killing in the short term, unlike the 4-hour time scale. But over like the 2-week, 3-week time scale, that's sort of that's a recipe for exhaustion, right?
Yeah. I'm thinking maybe tumor cell as a population, maybe they sacrifice some tumor cells become stiffer, then T cell keep killing them and they become quickly exhausted. Maybe right? That whole
that would be one way to leverage heterogeneity
just my guess. Last question. So for tumor cell, usually why they like the metastasis to the brain. Brain actually is much softer than lung, for example, right?
Yeah.
Why is that? They want to avoid the T cell killing, or for some reason they like to go to brain?
Well, so I think each microenvironment has its own challenges for the cancer cell. Joanne talks about this all the time, and so I mean the brain is a great place to grow if you want to get away from just sort of immune cells in general, right?
Right.
But yeah, I mean it's also you also have to, if you want to colonize the brain, you have to evolve other ways of anchoring to the matrix, other ways of proliferating in response to extracellular stimulation. So you're not going to be able to activate, like say, YAP signaling as well in the brain, right? Because you just don't have those. So as a metastatic cell that's colonizing the brain, you just sort of have to evolve a different set of strategies. But metastatic cells are great at evolving, and they
okay, I see.
Yeah.
Okay, thank you.
Thank you.
Thank you.
Great. So actually, before you go, Morgan, I have one more question.
Mm-hmm.
So the adaptation of the cancer cell regarding to the environment is really fascinating. I'm actually curious, do you have any insight regarding to the magnetic regulation of how cancer cell changes stiffness from the molecular level when they adapt the environment?
So, I mean, I guess there's been a fair amount of work done on this, just maybe not with this cancer cell specifically, but with sort of adherent cells in general and how they respond to environmental stiffness. Generally, you get enhanced integrin-mediated adhesion. And that generates signals, outside-in signals that move through the outside-in pathways, like sort of vinculin, paxillin, focal adhesion kinase, SARC, leading to then transcriptional stages that are driven by molecules like YAP to control differences in cell state, right? So myosin is very important. If you in the absence of myosin contractility, you don't get this same mechanosense. I think labs like Mike Sheets's lab. He used to be at Duke. Then he was at Singapore for a while, and I think now he's at Galveston. Has done a lot of the very foundational work on this topic.
I see. Okay. I do not see any further question in the chat box, so I don't want to keep it too long, Morgan, but thank you so much for the really inspiring seminar. We learned a lot. And then if anyone from the audience had the questions, just go ahead, shoot Morgan an email, ask him a question. I think it's really, really interesting work. Thank you so much. And hopefully we'll see each other soon, maybe sometime in person in NIH.
Hopefully.
Yeah.
Yeah, thanks so much for the invitation. And I really enjoyed the discussion. The questions were fantastic.
Thank you so much.
Thank you, Morgan.
Thank you.
Thanks.
Yeah. Bye-bye.
Bye-bye.